The species problem. The trouble with labels. Did you know that there is no scientific consensus to the definition of the word species? How in the world do scientists know if they're even talking about the same thing? I will try to clear this up, but it's gonna be tricky. Hold on to your seats. One of the issues is that when we find fossils, they don't have pretty little tags hanging off of them that says Homo heidelbergensis to let you know what it is that you have found. And since that is the case, humans take it upon themselves to label and categorize them. And this is what we call taxonomy. The trouble with taxonomy is scientists don't agree as to where one species begins and another ends. So the question then becomes, who gets to decide, and by what methodology do we organize mankind? Different cultures have different ways of organizing and structuring mankind according to their own perspectives, which makes the issue even more complicated. The species problem is considered one of the oldest, if not the oldest challenge in biology, which is the ongoing difficulty and disagreement among biologists in defining and identifying species. It's the challenge of finding a universally accepted definition of species that applies to all living organisms and reflects how they are classified. You would figure since the most familiar plants and animals we know are of different species, you would think there is a scientifically accepted method. So then the question obviously becomes, what is the method since we have all these different species? First off, let's deal with some of the definitions we do have. The 3 definitions of species. The word species can have different meanings, which sometimes overlap or even conflict with each other. Let's break them down. 1. Species as a taxonomic rank. This is like a label in a filing system. Species is just the name of a category used in scientific classification. For example, kingdom, Animalia, phylum, Chordata, class, Mammalia, order, primates, family, Hominidae, genus, Homo, species, Homo sapiens. In this sense, species is a rank, like family or genus. 2. Species as a specific taxon. Here, species refers to a particular group in the system, like Homo sapiens. It's not just a rank, it's about the actual organisms that are grouped together based on shared traits, like anatomy, behavior, and genetic similarities. For instance, consider the domestic dog, Canis lupus familiaris. It belongs to the species Canis lupus, which also includes wolves. This concept is about the actual organisms grouped together based on shared traits like anatomy, behavior, and genetic similarities. Wolves and dogs are considered the same species, but apes and humans are not. This can be confusing because the concept of a species is not always straightforward. While wolves and domestic dogs are considered the same species, Canis lupus, due to their ability to interbreed and produce fertile offspring, apes and humans are categorized into different species because they lack this interbreeding capability. However, the distinction is complicated by evolutionary relationships. Humans share a common ancestor with other great apes, such as chimpanzees, and the genetic differences between humans and apes are relatively small. This overlap in traits and genetic closeness often blurs the lines, leading to confusion about why some groups are considered the same species while others are not. The criteria for defining species can vary depending on whether the focus is on genetics, morphology, or evolutionary history, further adding to the complexity. 3. Species as an evolving group. This definition focuses on nature itself. A species is seen as a group of organisms that are evolving together over time. For example, consider Homo sapiens, the species to which all modern humans belong. Our evolutionary journey includes early human ancestors like Homo erectus and Homo habilis, who gradually adapted over millions of years, leading to the development of unique traits such as advanced tool usage, language, and complex social structures. This illustrates how humans, as part of an evolving group, have changed over time. The different species concepts. There are several different species concepts used by biologists to define and classify species. 1. Biological species concept. This concept defines a species as a group of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. In essence, it focuses on the ability of organisms to produce viable, fertile offspring together. 2. Morphological species concept. This concept defines species based on observable physical characteristics or morphology. Organisms that share a similar set of traits are classified as the same species, while those with distinct differences are considered separate species. This is often used when reproductive data is unavailable. 3. Ecological species concept. This concept defines species based on their ecological niche, which is the role and position a species occupies in its environment. Species are considered distinct if they occupy different ecological roles, even if they might be able to interbreed. 4. Phylogenetic species concept. This concept defines species as the smallest diagnosable group of organisms within which there is a parental pattern of ancestry and descent. It focuses on evolutionary history and shared ancestry, essentially identifying species as the tips of branches on the tree of life. 5. Other concepts. While the above are the most common, other concepts include the evolutionary species concept, which focuses on lineage and evolutionary history, and the cohesion species concept, which emphasizes the mechanisms that maintain the integrity of a species. There's also the phonetic species concept, which relies on overall similarity, and the cladistic species concept, which defines species based on shared derived characteristics. The choice of which species concept to use can depend on the type of organism being studied, the available data, and the specific research question. Sometimes, different concepts may lead to different classifications, which further complicates the issue. Anthropology and paleontology. One critical aspect of the species problem is the fluidity of genetic variation across populations. In biology, fluidity of genetic variation refers to the continuous and overlapping nature of genetic differences among groups of organisms. Rather than existing as sharply defined units, populations often share genes and exhibit gradual changes, making it difficult to draw clear boundaries between distinct species. Modern biology and genetics demonstrate that human populations, past and present, rarely exist in isolation. They intermix, share genetic material, and evolve in ways that defy rigid categorization. This makes it challenging to delineate species or subspecies, especially when relying on incomplete fossil records or genetic snapshots. One of the delineations or criteria for determining different species is that two distinct species are not supposed to be able to procreate and produce viable offsprings, meaning the children of the two separate species should not be able to reproduce. Neanderthals, Homo neanderthalensis, and modern humans, Homo sapiens, are often considered separate species. However, genetic evidence shows that they interbred, and many modern humans carry Neanderthal DNA. This interbreeding blurs the lines between species and challenges the notion of clear separations. Subspecies. The term subspecies is often used in anthropology to describe human populations with distinct physical or genetic traits. However, this classification can perpetuate misconceptions about human diversity, suggesting clear separations where none exist. For example, the classification of different human populations as subspecies based on physical traits can reinforce outdated and harmful notions of racial differences. A real-world example of how the term subspecies has perpetuated misconceptions can be seen in the historical misuse within early anthropology. In the mid-20th century, some researchers classified the indigenous populations of Australia as a subspecies separate from other humans. This was based on superficial physical traits, rather than rigorous scientific evidence. Such classifications not only lacked a foundation in genetics, but also fueled discriminatory practices and policies. Modern science, through extensive genetic research, has since shown that indigenous Australians share deep genetic ties with other human populations, further debunking the validity of such outdated classifications and asserting our shared human ancestry. The concept of subspecies in modern humans is generally not supported by genetic evidence. Variability among human groups is gradual and overlapping, a pattern known as clinal variation. Clinal variation refers to the gradual change in certain traits or genetic characteristics across geographic areas or populations, rather than abrupt differences between distinct groups. Instead of clear boundaries, traits such as skin color, height, or genetic markers shift slowly and continuously as you travel from one region to another. For example, you might notice that skin pigmentation becomes progressively lighter the farther populations live from the equator. This smooth gradient reflects how populations adapt to local environments over time and illustrates why humans cannot be neatly divided into sharply separated biological groups. In essence, clinal variation highlights the continuity and interconnectedness of human diversity. The genetic differences that exist are far less significant than those that would be found between true subspecies in other animals. In the scientific community, the use of subspecies to describe human populations has been largely abandoned because it does not accurately reflect our evolutionary history or genetic reality. Incomplete fossil records. Fossil records are often incomplete, making it difficult to draw clear lines between species. For instance, the classification of early human ancestors like Homo habilis is based on limited fossil evidence, leading to debates about whether they should be considered separate species or not, considering the lack of fossil evidence. Reconstructing entire skeletons and hypothesizing about the life of ancient organisms based on minimal fossil evidence is a fascinating, albeit speculative, process. Paleoanthropologists and paleontologists often rely on fragmented fossils, sometimes just a single skull or a few bones, to infer the complete anatomy, behavior, and ecological roles of extinct species. Here are a few notable examples of this process. Homo habilis. One of the earliest members of the genus Homo habilis, is reconstructed primarily from scattered cranial and postcranial fossils. While only partial skeletons have been unearthed, scientists use comparative anatomy by studying existing primate species and other hominin fossils to fill in the gaps. For instance, a fragmentary jawbone is extrapolated to estimate the shape of the skull and facial features. The tools found near fossils often provide clues about their intelligence and lifestyle, suggesting Homo habilis was a toolmaker and scavenger, living in a semi-nomadic social structure. Homo erectus. The famous Turkana boy, a nearly complete Homo erectus skeleton found in Kenya, is an exception to the rule, but far more commonly, reconstructions of Homo erectus rely on piecing together isolated bones discovered across various sites. Combining these fragments with measurements and digital modeling allows scientists to theorize about their upright posture, robust build, and endurance-based hunting techniques. Reconstruction suggests they lived in coordinated groups, developed fire usage, and migrated extensively. Australopithecus afarensis, Lucy. Lucy, one of the most famous Australopithecus specimens, consists of only about 40% of a complete skeleton. Scientists use mathematical modeling and comparative anatomy to infer her height, gait, and physical appearance. Further studies of the surrounding environment suggest she lived in a mix of savannas and wooded areas, capable of both climbing trees and walking upright, a dual adaptation that hints at transitional evolution. Sahelanthropus chadensis. The discovery of Sahelanthropus chadensis in Chad in 2001 marks another instance where scientists created a vision of an ancient hominin based on limited evidence. The entire reconstruction was derived from a single skull, albeit a nearly complete one, dating back approximately 7 million years. Despite the scarcity of postcranial elements, researchers have speculated on Tumai's ecological niche in locomotion, blending insights from contemporary primates and other early hominin fossils. Kenyanthropus platyops. Unearthed in Kenya in 1999, Kenyanthropus platyops is another example of a species reconstructed from a single, highly fragmented skull. Dating back roughly 3.5 million years, this hominin displayed unique traits such as a relatively flat face, which set it apart from contemporaries like Australopithecus afarensis. The reconstruction process involved piecing together the shattered skull fragments to hypothesize about cranial morphology and facial structure, supported by comparisons to other fossils from the same era. Paranthropus robustus. First identified in South Africa in 1938, Paranthropus robustus was reconstructed from partial skull fragments and isolated teeth found at various sites. This species is characterized by its robust cranial features, including a pronounced sagittal crest and large teeth adapted for heavy chewing. Despite the lack of complete skeletons, Scientists have inferred body proportions and dietary adaptations by comparing these fragments to other specimens in the genus Paranthropus. Piltdown Man fraud. The Piltdown Man was a famous paleoanthropological hoax in which fragments of a skull and jawbone were presented as the fossilized remains of a previously unknown early human. Discovered in 1912 in England, the Piltdown Man was believed to be the missing link between apes and humans. However, in 1953, it was revealed that the Piltdown Man was a forgery, consisting of a modern human skull and the jawbone of an orangutan, artificially aged to appear ancient. This fraud misled the scientific community for decades and highlights the potential for deception in the field of paleoanthropology. The examples above underscore the ingenuity of paleoanthropologists in reconstructing entire species from minimal evidence. Each fossil provides a snapshot that, when combined with modern technology and scientific inference, opens a window to the distant past. Beyond anatomy, artists play a crucial role in visualizing the lives of these ancient organisms. By collaborating with scientists, artists create representations that postulate skin texture, coloration, and musculature based on modern analogs and fossil evidence. These reconstructions often depict the organisms in their habitats, offering a glimpse into their diets, social behavior, and environmental interactions. Genetics. The advent of advanced genomic sequencing has made it easier to trace evolutionary paths and migration patterns, but has also led to oversimplifications in popular science narratives. How we use these tools matters, whether to foster understanding or perpetuate divisive myths. Genetics can offer powerful insights into the relationships between organisms and help clarify whether groups should be considered separate species. By comparing DNA sequences, scientists can estimate how closely related different populations are and trace patterns of ancestry and divergence. In many cases, significant genetic differences may support the classification of separate species, while high genetic similarity might suggest a single species with variation among populations. However, the picture is rarely that straightforward. Gene flow, interbreeding between populations, can blur boundaries even between groups that appear quite distinct physically. This is especially evident in the case of humans and Neanderthals. Genetic evidence shows they interbred, sharing portions of their genomes, even though they are usually classified as separate species. Furthermore, genetic differences do not always align neatly with physical or behavioral distinctions, and the amount of divergence required to warrant species status can be somewhat arbitrary and dependent on which genes are examined. Additionally, when fossil evidence is sparse and genetic material is hard to recover, as with many ancient remains, scientists are often left to piece together evolutionary relationships from fragments. In summary, while genetics provides invaluable clues, it is only one part of a larger puzzle that includes anatomy, behavior, ecological roles, and evolutionary history. The decision to classify groups as separate species remains complex, often requiring a combination of evidence from multiple fields. Another layer of complexity arises when discussing jargon such as non-human DNA or non-African DNA. These terms, though scientifically intended to describe genetic contributions from other hominin ancestors like Neanderthals or Denisovans, can unintentionally evoke a sense of otherness or hierarchical difference. The framing risks reinforcing outdated notions of purity or superiority, which are at odds with the reality that these genetic components are part of our shared human tapestry. Using these terms can confuse the layperson to believe that people with non-African DNA don't descend from the so-called African, or that non-human DNA, in terms of human ancestry, refers to something other than archaic humans. Let's give a little more context. Non-human DNA and human genomes. It is scientifically accurate that modern humans have inherited small segments of DNA from archaic human species, such as Neanderthals or Denisovans, due to interbreeding tens of thousands of years ago. These segments are often colloquially described as non-human or archaic DNA, but it is important to clarify that Neanderthals and Denisovans are human relatives, not completely non-human entities. The phrase non-human DNA could be misleading if interpreted outside this specific scientific context. Non-African DNA and human ancestry in population genetics Non-African sometimes refers to genetic lineages or variants more commonly found outside Africa, often due to historical migration patterns. All modern humans have common ancestry originating in Africa. The presence of so-called non-African DNA segments, like Neanderthal DNA, outside Africa, reflects the historical interbreeding events after humans migrated out of Africa. This terminology does not imply that any population is not descended from African ancestors, but rather highlights differences in archaic admixture due to migration and interbreeding. The concern that these terms can reinforce outdated notions of purity or create false ideas about genetic superiority is well-founded. Scientific language, when not carefully explained, may inadvertently perpetuate misconceptions about human diversity and ancestry. Persistent confusion stemming from the species problem. Unlike elements in chemistry and particles in physics, biology's best-known unit, the species, remains ill-defined and disputed. Although it continues to be used routinely by researchers from across the field of biology, there are at least 30 competing definitions termed species concepts in existence. Ongoing discussion about the limitations of one of the most famous species definitions, the biological species concept, highlights the continued difficulty of resolving this species problem. Its emphasis on reproductive isolation means the biological species concept remains notoriously difficult to test empirically and cannot be applied to asexual organisms, including prokaryotes. Worse, even its proponents admit that notions of RI are undermined by evidence of widespread hybridization between otherwise distinct species, as well as by the protracted and variable nature of speciation itself. Many biologists therefore choose to use other definitions that emphasize criteria such as mate recognition, ecological niche, or monophily, but incompatibility among these competing definitions means that this pluralism in relation to species concepts can lead to differing conclusions about species delimitations and counts. This species problem has ramifications beyond taxonomy because an indefinable unit is a potentially incomparable one. Knowing which species an organism belongs to is integral to studying and understanding both that organism and the wider world. But if different researchers are using inconsistent definitions of the term species, the comparability of their results might be brought into question. Since species form the basic unit in research as diverse as understanding speciation, estimating biodiversity, modeling species distributions, and conducting conservation assessments, inconsistency in their delimitation impacts biologists from a wide range of disciplines, such as systematics, evolutionary biology, ecology, and conservation. Ethnocentric bias, the ethnocentric lens in taxonomy and genetics, further complicates these challenges. Western scientific traditions have historically dominated the narrative, imposing their classification systems onto other civilizations' understandings of human variation. Indigenous frameworks for identifying and categorizing people, often based on lived experience and environmental adaptation, are sidelined, despite their validity. These systems deserve exploration, not as quaint curiosities, but as contributions to a holistic understanding of humanity. The tendency to interpret and judge other cultures by the standards of one's own has left an indelible mark on the scientific enterprise of taxonomy and the classification of human diversity. Across times and places, classification systems have functioned as mirrors, reflecting the worldviews and prejudices of those who build them. Whether through Western racial science, imperial census categories, or traditional social hierarchies, humans have repeatedly constructed boundaries that favor their own norms and diminish the other. Recognizing this history is essential to moving toward more inclusive and scientifically rigorous understandings of human diversity. Blurred lines, the human continuum, perceptually different, inherently the same. The arbitrary nature of boundaries between types of humans highlights not only the subjective standards set by scholars, but also the broader philosophical question: what does it mean to be human? Evolution is continuous, and the distinctions we draw often reflect societal and cultural biases rather than biological imperatives. Genetic markers may indicate shared ancestry, but they often fail to encapsulate the cultural, historical, and personal significance of how individuals and communities define themselves. In conclusion, the species problem remains one of the most enduring and complex challenges in biology. The lack of a universally accepted definition of species highlights the fluidity and diversity of life on Earth. Different species concepts, such as the biological, morphological, ecological, and phylogenetic, each offer unique perspectives but also contribute to the ongoing debate. The interplay between genetics, fossil records, and cultural biases further complicates our understanding of species boundaries. As we continue to explore and classify the natural world, it is essential to recognize the limitations and subjectivity inherent in our definitions. By embracing a holistic and inclusive approach, we can better appreciate the rich tapestry of life and the interconnectedness of all living organisms.
Sometimes I don't know what to say. It's a genuine miracle I woke up today, so I got up to pray. But my BBM was pinging when my Android started singing. Then I missed all of the glory for technological luxuries. And just like that, I forgot all of the trees and the flowers in the breeze carrying seeds across the seas. Extra honey in my tea, but pay no homage to the bee. Whatever happened to us? And will we ever let the magic come and tap into us? We preach apocalypse written by John the Revelator, but won't even speak to a stranger riding on the elevator, or step to the side when we standing still on the escalator. The planet Earth is a hospital, we on the respirator. I don't regret the haters. Sometimes that's what you need to see yourself, break through and free yourself, accept your own and be yourself is magic. The story of life is too tragic.